directly impact beer and brewing quality. So today we're going to be talking about understanding protein and understanding kind of the challenges of high protein barley, as well as some of the breeding strategies to kind of suppress barley synthesis, or protein synthesis, but really promoting starch synthesis. So excited to have both our speakers here today. We have Dr. Glenn Fox, the Anheuser-Busch Endowed Professor of Malting and Brewing Science at University of California, Davis. And we have Dr. Jamie Sherman, Associate Professor and Barley Breeder at Montana State University. So Glenn is going to kick us off talking a little bit more background about protein and different proteins that build up, that make up barley structure. And then Jamie's going to talk a lot about her research and working on the extended grain fill trait, promoting starch synthesis, especially in dryland agriculture. So very excited for both of those. We're going to go through both of the presentations and then do Q&A at the end. So if you do have questions, please type them in the chat and we'll be pulling them out at the end. But we'll also just kind of open it up to the audience as well. If you have any technical difficulties, shoot a message to Wade or me and we'll get you sorted out. Otherwise, I'm going to hand it off to Glenn and we'll get started. So thank you.
Thank you, Campbell and Wade, for the assistance here. Good morning, everyone. Maybe good afternoon for some folks. So I'm going to sort of give an introductory presentation around protein itself. And we're sort of covering specifically free amino nitrogen development. And while it's saying on beer quality, there's an understanding that would assume it's also associated with malt quality. I'm hoping most people are familiar with barley accreditation. So there's a protein content window that malting barley is accepted at. And the example we could look at, say, we've got a sample at 12% protein, but we know that not every grain in that bag of barley or that truck of barley is 12% protein. There's significant variation. Farmers will use things like yield maps, and we can also associate protein maps and understand variation across a field. And that example in that image on the bottom left, there's nice lumpy little field, and you can imagine that there'd be slight variation in maybe the nutrition profile in that field. So we expect some inherent variation in this sample. And a long time ago, I had a student who still talks to me, believe it or not, where I got her to measure a couple of thousand kernels individually for protein content. And this was the variation we saw in a sample. So even this one sample here was significant variation in protein content, but it averaged 12%. So we have this inherent total amount of protein variation, but is it only total protein we should be really focused on, or are there other aspects of protein we could consider? So some of you may have been unlucky enough. I know I can remember doing Kjeldahl analysis a long time ago, boiling sulfuric acid and then boiling caustic to do the titration, to do the distillation. Well, Kjeldahl was working at Carlsberg laboratory, and some of his equipment is still there. If you go visit Carlsberg and go through the museum, some of his equipment is still there. And then a Frenchman, Dumas, came up with a combustion method, which is the more common method used today. But both of these are measuring total protein. Probably around 40, 50 years ago, we in terms of the royal we, discovered ways to separate proteins based on either size or charge of proteins. And they were using 1 or 2 dimensional electrophoresis to really separate individual proteins, look at proteins in abundance. And people might be familiar with using SDS page, that bottom image on the left, 1-dimensional electrophoresis, where we could do varietal identification. And usually the B Hordines were the most useful in doing varietal identification because most varieties at the time were still unique. We've had a lot of interbreeding and crossbreeding and parentals used now, so sometimes it's more difficult to use electrophoresis as varietal identification. If we separate on size and charge, then we can actually see individual proteins. So we go from looking at maybe a dozen proteins in a lane on that SDS gel to looking at maybe 40 or 50 proteins in total. And then came along proteomics, which really changed the game and how we could understand every protein that might be present. That's using enzymes. So we're actually only identifying peptides from proteins, and those peptides are associated with proteins. But again, it's the same principle that we're interested in understanding protein digestion. When we talk about total protein? Well, this is just an example based on the Osborne fractionations that Osborne developed over 100 years ago, separating proteins based on their solubility. So the albumins and globulins were soluble in water. The globulins, you need a little bit of salt. Then we could separate proteins on using alcohol, and sometimes we needed alcohol with a reducing agent like smelly dithiothiazole or something, mercaptoethanol, very, very smelly stuff. But we can see some common proteins in this list here. We'd be familiar with beta amylase, LTP, a foam positive protein, photoindolines, which impact structure, serine protease inhibitor, again, a foam positive protein. And of course, our storage proteins over here are hordenes. Now, the analogy is these are also the same proteins in wheat. There's storage proteins in the other grains, but there's a slight difference in the amino acid sequence that makes the wheat and barley proteins have special properties. If we're talking specifically about the endosperm storage proteins in barley, and again, think about wheat as the homologue here, this lovely SEM image here, and I love to have students spend quite a bit of time looking at this image and try and decipher what this image is really all about. We do see some standouts here and some lovely big starch granules. And then we see lots of tiny little small starch granules that are packed around in this matrix. And this is actually the storage protein itself. You can see it can be quite webbed, it's packed really tightly. And this is where the small granules are packed really within the matrix itself, whereas the large granules might only be having a small surface area that's interacting with the Hordine matrix. Now, if we talk about how much of this makes up total protein, well, it can range between 40 to 70% of total protein is this storage protein. Now, that's a big difference. While we can say, oh, it averages around 50, 55%, we've tested samples that are in the low 40% and samples in the high 70%. Now, that relationship does impact on total protein content as well, or is impacted by total protein content. So if you have a sample of 12% barley, you could have 50 or 60% storage protein. You think, well, is that a big difference between 50 and 60%? It depends, but it's still different. You could have samples at the same protein content, but different amounts of storage protein. And we have found that those different amounts of storage protein do impact malting quality in particular. The other thing that's important to understand about this, we might think, well, this is all the hordenes themselves, but there's other enzymes, other proteins bound up in this matrix. Beta amylase, the bound up beta amylase, is hiding in this matrix. Serine protease inhibitor is also bound up in this Hordine matrix. And the photoindolines are specific proteins that we hypothesize are actually binding the protein to the granules. So while we think, oh, okay, we've got 40 to 70%, which is a significant amount of total protein, higher levels of this impact texture. And harder texture can mean it's slightly more or a longer process to modify the malt. It also might have impacts on milling efficiency in the brewery. So while we think we've got barley within the desirable protein window, the storage protein is potentially a hidden problem that we don't quite know, or we don't know, but there still could be some issues. So it could impact hydration, as I mentioned. It could impact milling, and certainly it will impact gelatinization and how these starch granules can swell. So all of these things suddenly are potentially being impacted by the amount of storage protein, not necessarily by the amount of total protein. The other point I'll make about particularly the wheat and barley proteins is this high abundance of our little friend proline. Now, we could have a pop quiz. We do call proline an amino acid, but technically it's not. It's an amino acid because this amine group is bonded. It almost looks like valine, but it's not because this is bonded. So this amine group is not available. So technically it's amino acid. It's a great biochemistry question for the students. Our proteins are really rich in this particular amino acid. And the concerns are usually that yeast is not assimilated, does not assimilate the proline. So there's lots of proline left over in finished beer. And one of the problems is that proline can bind with polyphenols to cause haze in beer. That's going before we really wanted to make hazy beers. This is in the days where we talked about bright beer. Now we talk about hazy beers. And this is one of the reasons we use protein or wheat proteins to make hazy beers. Comparing SDS page and proteomics, again, looking at composition. So this is an example of an SDS page gel from Christine Finney over there in Carlsberg, and looking at these D-hordenes, some C-hordenes, and the slightly lower molecular weight, but more abundant in terms of numbers of B-hordenes. And then we have way down here some gamma-hordenes and even smaller proteins. If we do proteomics, and we can actually look this just a single variety, UC Capet, grown over 10 locations or so. This is a few years ago across multiple locations and multiple sites. These are abundance numbers. This is not quantitative. These are abundance numbers. And technically, we only look down the column. We shouldn't compare across, but yeah, we do. So we can just see beta amylase, for an example, significant difference in the total abundance, even with samples that have a very similar protein content. So the amount of this protein changes, whereas the total protein content doesn't. And that's driven by other proteins that are going up or down in abundance. And we can see here with the large D-hordene protein, which is usually having a major negative impact on malting and brewing quality. It really does has a negative impact on extract development. It's the big guy. And even two samples here at 10% protein, almost double the amount of D-hordene. We can look down here as well and see the same sort of differences between these subunits of the B and C hordenes. And then we have our hordoindolines, the A and B, and they certainly impact texture. So using proteomics, we can start to understand the potential impact that these might have, but we know how time-consuming it is to get proteomics. It's not an immediate turnaround in the lab, and usually it can take more time analyzing the data than it can to actually get the data. If we talk about how it relates to quality, if we talk about malting in particular, and this has probably been one of the holy grails for our industry for a very, very long time. I know Bernie Jones worked on this for a long time when he was at Madison, and then we had some colleagues in Australia do similar research over a long time, and they were trying to get a more robust enzyme methods working to quantify the enzymes that would hydrolyze hordenes and quantify the amount of hordenes. It was difficult, and in the end, Bernie and others said this is really hard, and this is going to take probably 20 or 50 PhDs over a few decades. So it sort of has died in interest or lack of interest at the moment, but this is where proteomics may come in and help resolve some of our understanding of the enzymes. So on the left graph here, we have the development of three enzymes, three proteases. One is already bound up in the barley itself. This is time of sampling over a 7-day malting process. We see the drop when they go in the kiln. But at the same time, we see changes in abundance of our storage proteins. So this is where we might be able to start to put together a bit more of a more detailed picture of what's going on with protein hydrolysis. Now, this is the important part of what we're wanting to do. We're trying to modify the storage protein. So enzymes will have access to the starch granules, but also we're wanting to produce some free amino nitrogen. But we don't want to over-modify our malt, because then some of the peptides that are responsible for beer foam might not be the type of structure or the length of the peptides we need for beer foam. So this is where modification starts to become part of the understanding of why we want to do what we want to do and understand storage proteins. And these are the peptidases or the proteases we talk about. This is the pH range in this left column. This is optimal temperature, and then this is potential degradation temperatures or denaturation temperatures. You can see here that only a couple really work in the system we work in. at that lower pH range, usually can work well in a low mashing temperature system. And there's only really one carboxypeptidase that is quite thermostable. So again, becomes important when we're considering mashing protocols. There are four classes of proteases, but there are more than four proteases. And the magic number we think is 42. The cysteine proteases, there's one particular one, this EP1, or was called MEP1 back in the 1980s. We believe this is responsible for releasing beta amylase from the Hordine matrix. But there are other cysteine proteases as well. The two main proteases we think about are the serine proteases, the chymotrypsin and trypsin. These are also used in proteomics. So these are more common enzymes when we're using proteomics to digest all the proteins. There's that aspartate protease that's bound up in the barley itself. And then we have some carboxypeptidases. Now, I'll make a point of the trypsin and chymotrypsin because these are ubiquitous proteases in most living organisms. So as you're digesting protein in your stomach and intestine, it's these proteins here. So just as an example, here's one of the B Hordines. This is the amino acid sequence itself. This is not a full amino acid sequence. I think it's only around 300 or 400 amino acids. What I'm trying to demonstrate here is the abundance of P's and Q's, glutamine and proline. These two amino acids, particularly in these rich motifs of repeated sequence, are what celiacs respond to or causes the celiac condition. And wheat and barley are abundant in these P and Q motifs. If we were looking to hydrolyze these proteins, then we can use an example of one trypsin enzyme, and that amino acid, or that particular protein, would be digested into about 8 different fragments. We do have one really long peptide here of 142 amino acids. And again, if we look at the P's and Q's, okay, this is still quite rich in the P's and Q's. There's smaller peptides here, and still a couple of relatively long ones as well. If we look at that really long peptide with 142 amino acids, and we think, oh, that's all very nice, but we know some of these peptides contribute to foam properties. If we then look at the hydrophobic amino acids, these are the amino acids that theoretically they're trying to escape. They don't like being in water. So they are part of the foam properties, the foam wall, the bubble walls. So these yellow amino acids are the hydrophobic amino acids. So when we have really rich portions of these long proline and phenylalanine and some other amino acids, then these become our foam positive peptides. Where do our free amino nitrogens come from? Well, the carboxylic acid or the carboxypeptidase is slowly munching along the end of this chain here from the carboxy end. And it'll just release a single amino acid until it gets to something like proline. And it's not a stopping point, but it's a slowing point. The carboxypeptidase can get around this bond between the proline and its adjacent amino acid, but it slows the process. But this is where our free amino acids are coming from. If we jump back up here and look at the starting point where these are working from, we can actually see that we'd get a lot of amino acids released before maybe it got to a proline that could slow the point, slow its continued hydrolysis. So this is where our amino acids are coming from. So we have our peptides being released from the large protein. We send up with these small fragments. Some of these fragments are quite rich in hydrophobic amino acids, so they contribute to beer foam. And then we have the free amino nitrogen being released from them as well. Why do we care about free aminos? Well, hopefully we have a good understanding that yeast requires amino acids for as a nitrogen source. It will assimilate amino acids based on preference. We have our A, B, and C options, and theoretically, again, proline is left out. It is not assimilated, but of course, yeast will make its own proline. You cannot make any protein unless you have some proline. So the yeast is making its own proline. It's not terribly relevant. You know, for today's conversation around the difference in those classes, other than the yeast will consume everything most of the time except proline. We only think about amino acids in beer coming from our malt sources, but hops also contribute some amino acids because they have proteins as well. So this particular figure here from some work a former student of mine did when he was working on a Hop Creek project, the difference between hop amino acids and some malt amino acids. And again, we see this rich abundance of glutamine and then proline, whereas in hops, it's more like it's asparagine. That's a dominant amino acid. Why do we care about amino acids? Well, amino acids, other than being a nitrogen source for yeast, are potential off flavors or precursors to off flavors. And we've tested a couple of hundred beers the last couple of years, and we're still testing more. And I've got students in the lab at the moment testing a bunch of beers, and we've got 200 on the shelf that we're testing at the moment. And these are some. This is double IPA. This is some hop water. It's an IPA, some kombucha, a lager, a pale ale, some sour beers, and some triple IPAs. This is the average fan we're finding across a range of beers. This is a couple of hundred beers, varying in the number for each one we've tested, but we're seeing some really significant total amino acid values in finished beer. Is this a problem? Potentially because it's a problem for potential precursors for off flavors. Now, when we do these tests, we don't know the amino acid profile. We're just saying this is the total amount of amino acids, not including proline. Then we see some others that are really low, and 40 to 50, I think, for most lagers would be, so that's the preference, and most ales, it's certainly less than 100. So we are seeing some beer styles that are really potentially problematic in having way too much free amino nitrogen. We also know that the storage proteins themselves impact gelatinization because the granules, starch granules, have to swell. And then as a consequence, that might impact total fermentable sugar profile. So here's our B, D, C, and A, and having a negative impact on most of the fermentable sugar profiles that we're looking for. So again, this is how the total storage protein is impacting on several aspects of both malt and beer quality. And I've gone too quickly. So just in summary, while we're talking about protein, I think we can't talk about protein in isolation. And in this particular presentation, I've considered starch as well. I think we have to move from this content paradigm into a composition paradigm as well. Having said that, there are some challenges with that. We know that total amount of protein and total amount of storage protein can impact modification. Gelatinization can impact brewing efficiency and can impact final beer quality. Now, people have asked me when I've given similar presentations and talked about storage proteins, can we measure total storage protein? Yes, we can. We've got an NIR calibration. We can measure this. The slow point there is currently the calibration is only developed for ground grain, and it's only barley. So it is a slowing step. We can't just scan our whole grain. We have to scan milled barley. But again, we're getting good information on the variation in storage protein in samples. I think this would be of great future benefit for maltsters in understanding, potentially how they're going to be processing that grain, but also then being able to have conversation with brewers about what the malt might do in terms of these other things in beer quality. So Campbell, hopefully that wasn't too long or maybe too short.
No, that was perfect, Glenn. You hit it with 1 minute to spare. So thank you. I'm going to pass it on to Jamie, who's going to talk a little bit more about breeding strategies and how we can handle this in the lab in the field.
Just working on sharing my screen. Can you see the presentation?
Yep, thanks.
Okay, so let me try to move this. Okay, so I'm gonna jump in with the challenges of high protein kind of from a very different perspective. I would say more from the grower's perspective. When I first started working in barley, it was a big concern as far as the level of protein. And it's because the growers of Montana often have their malt rejected because the protein is too high. And so we were working towards developing lower grain protein. In a lot of regions, the grower can manage for lower grain protein using irrigation, but 80% of the acres in Montana are dry land, and it's a beautiful place to live, but usually during grain fill, we don't get a lot of moisture. And more recently, we've been having extreme heat events during grain fill, which cause a huge issue with grain protein. So, what are the causes of high protein in barley? It's these effects, but also an interaction between all of these. So you can choose a variety that tends to have lower protein, but even if you have a good variety, drought and heat and mismanagement of nitrogen is going to drive the protein up. And so, as we were working on this, we found that extending grain fill in, at least in Montana's very dry, hot grain fillet climates, can reduce the negative impact of the environment. Now, this was a little counterintuitive to me because it seems like by extending grain fill, you'd think that it would have a greater chance of being exposed to drought and heat events, but we have a lot of data to show that it does help. So by extending grain fill, what I'm talking about is from two directions. So you can have earlier heading, which means that you'd have a longer grain fill as long as your maturity is not also earlier. And then later maturity as well, you can extend grain fill that way. And so there's to get the longest grain fill, of course, it would be an advantage to have earlier heading and later maturity. Um, it tends that the heading and maturity usually correlate. So the later the heading, the later maturity usually. But we found some gene regions that break that association where you can have earlier heading and later maturity. And so we see that by extending grain fill, we have a number of positive effects. So we can see, so the x-axis is grain fill duration, and then we can see that longer grain fill can mean greater yield, can mean larger seed, greater number of plumps, can mean a lower grain protein, and can mean a higher malt extract. Um, and through our earlier work, we've identified four gene regions. I think there's probably a number of others, but in our material at least, there's four gene regions related to extended grain fill, and I'm going to talk a little bit more about those. Where we identified those is in this Williams paper published in 24. And the interesting thing is that they're all related to extended grain fill, but they can have different directions of effect. So, for example, the 2H one is earlier heading. The 6H gene region is later maturity. The first 7H one is also earlier heading, and then the last 7H is both earlier heading and later maturity. And so all of these extend grain fill in different ways, but they also impact other traits. So, for example, here you have the 6H allele decreasing protein, and and the 7H allele as well. The extended grain fill allele comes from two different sources in these two cases. 6H comes from the North Dakota line we were using, and then the 7H allele that extends grain fill happens to come from the Montana line. But they both decrease protein. Interestingly, they have a greater effect in the more easy environment in Bozeman, cooler, wetter. We see a bigger difference in protein than in the drier, hotter environment of Havre. But you can still have important differences there. Um, so another thing that we see with these gene regions is an association with plumps and because of that, then extract. And in this case, we tend to see a bigger difference in plumps at these gene regions in the hotter and drier environment of Havre. So, so the different gene regions have different sorts of effects, and the effects can be different depending on the environments. But interestingly, if you put them all together, so if you have all four gene regions with the extended grain fill alleles, then you're going to have your longest grain fill and, for example, in the other panel, the lowest grain protein, so the biggest effect on grain protein. And so, looking at it this way, there could be somewhat of a targeting. One of the concerns about extending grain fill, especially if you make it about a much later maturity, is that it could be so late that a grower can't get it out of the field. And there's been reports of this issue. Rich Horsley, who I got this material from, essentially abandoned it because it was too late for his growers. So in areas where you have plenty of water or irrigation, you don't have the climate like Montana where we have this heat and drought ending grain fill. Um, it's not a good idea because uh, maturity can become too late. In our case, in some of the stuff we've been doing, it hasn't changed harvest date that much, and it's just because of our environment. So these traits are to help specifically for hot and dry environments during grain fill. So like I said, when we first started, we were trying to solve some problems. Hocket had been accepted and was being widely used. It was high yield. It has stable plumps in dry land, but its protein can go above what's accepted and it tends to modify slowly. So we back crossed a single gene into Hocket, which HVNAM1, which is at the same locus on 6H that I've been talking about. And what it does is it delays maturity. It's a transcription factor that is mutated, so it does not turn on the normal senescence. So maturity is delayed, allowing more time for grain fill. And we've been calling this the low protein gene because the longer the grain fill, the lower the grain protein. And so we moved this back crossing and then we released this line called Buzz. It carries not only the 6H stay green, but also the early heading, which I think is key to its success. So it has both. And so higher plumps, lower grain protein, and higher extract are kind of the key traits that we're seeing there with Buzz. And then we did the same thing with another variety. We crossed hock it with another North Dakota line, which has extended grain fill. So it has the HVNAM1 6H low protein gene, but then some of those other uh, genes. And in the case of endurance, it ended up with the low protein 6H and the extended grain fill on 7H. And you can see that it has low protein, um, but its kind of superpower, I don't think is related necessarily to these two stay green loci. I think it's something else that we don't know what the gene is. Endurance has really large seed. And because of that, it's a good thing, and it's also maybe it's a weakness too, and under certain circumstances. The big seed meat protects it um, under dry land and drought conditions. It means it can still have really high extract. So you can see that its extract is much higher when compared on dry land to the controls. And it's just because of the really big seed. Now, the disadvantage of that is on irrigated, the seed is really, really big. And so then it can be very slow to malt. And so you would not want to grow endurance on irrigated ground. Oh, okay. So at the beginning, I said one of the things that can go wrong as far as protein is nitrogen. And so this is kind of a normal year in Bozeman 2023. You can see that we had four different nitrogen treatments, and you can see kind of a general effect on these different traits. So grain fill is increased with higher nitrogen. Yield goes up, but then plateaus, and then can even be negatively impacted if you go too high. Um, Seed size normally is negatively impacted if you go too high with nitrogen, and protein goes up with increased nitrogen. That dark black line is buzz, so you can see that it's relatively stable across all these nitrogen treatments. And I think because it's, you can see the yield is more plateaued. Hockett had a bigger negative impact of the nitrogen on yield. So I think Buzz is just more stable. It maybe could take more nitrogen and still have a good protein and plumps. So that's kind of a normal year, but it gets worse if you got hot and dry. So now grain fill has been cut back, 10 to 20 days, and plumps are decreased. And so you can just see now as we apply too much nitrogen, we have an even harsher effect on some of these varieties. And so that interplay between the conditions, the environment, and the nitrogen application is just really shown here. And so with the plumps, for example, buzz is even the plumps are below what you would want under the 1X nitrogen treatment in 2022. But its protein was still acceptable. So another way to think about this is just whether it meets AMBA specs or not. So this was a drought year in Conrad where we had these varieties under these different nitrogen treatments, and you can see that we're still below the 13% threshold for protein, and for the most part, we can stay with acceptable plumps with those two extended grainville varieties. Another interesting thing, if you look at these traits based on the gene regions, they have interesting different effects. So one of the things that we've become more concerned about is not just a problem with lack of water, but heat. And we found that in some of our extreme conditions, you cannot, even watering, if it's hot enough, you cannot fix the heat effects. So you can still have damage of heat even under irrigation. And so we're really interested in knowing more about heat resistance. And so we did some heat studies with these different gene regions. And you can see that as far as grain protein is concerned, several of the gene regions, if you have the extended grain fill, which is the green bars, have lower grain protein. Now, interestingly, the 2H one, which is the earlier heading one, had a little bit higher grain protein. And we were wondering if this was just some sort of oddity of the test or something like that when we applied the heat. But we're seeing a similar effect in a field study with this same locus where we had different nitrogen treatments. Actually, this is data from a greenhouse study, but different nitrogen treatments where we can see an increase in grain protein with that extended grain fill, which is the ND allele. Whereas the 6H gene region tends to have lower grain protein no matter what the treatment is. So that's interesting. And it's interesting not so much from a perspective of malting and brewing where you want low grade protein, but from a nitrogen use efficiency. So if we had this gene, could we apply even less nitrogen and it still get a good product? So that's something that we're continuing to explore. Okay, let's just talk a little bit about quality and the relationship between these different extended grain fill gene regions with quality. Here I just list the quality traits that were significantly impacted by these gene regions. So notice extract is at least with the first two gene regions relatively consistently impacted across environments where the stay green had a higher extract. We see alpha amylase tends to be higher. Uh, Soluble to total protein tends to be higher, and so soluble protein is higher. So I think what this is telling us is similar to what was previously discussed that it's a modification issue where for some reason these stay green lines modify better. Note that the other two gene regions don't have very significant associations with these quality traits. And you might say, well, why don't we see FAN in this? And FAN's relationship seems to be a lot more inconsistent, but I did pull up some new data that I thought you might be interested in. And granted, may not be significant, but there's definitely some interesting trends here where FAN is going up with the stay green or trending up with the stay green allele. So in these graphs, the non-stay green allele is listed first. There are three different years, and I had to separate the data out by year because when I didn't, you kind of lost all the differences. Um, and so environment is really important here. But there does seem to be a trend where the stay green has higher FAN in with the 2H allele, one of the ones that impacted protein, and then the 6H as well. And then when I put the two together, so if I had the non-stay green allele for both, that had the lowest FAN. If I had the stay green allele for both, then I had the highest FAN. So that's what I can say about FAN. Unfortunately, we haven't done anything to look at differences in composition. Um, why are these lower protein? Is it storage protein? Is it one of the other proteins that was talked about previously? So that would be a really cool thing to investigate further is what is really the difference here? And that's really all I have for you. We'd be happy to take any questions.
Amazing. Thank you both to Jamie and Glenn. I think this is a really important conversation, both just as we as an industry still struggle to understand protein content, amino acid composition going into our word streams, but also seeing some really innovative genetic tools to kind of start tackling this and thinking about kind of more unique growing regions and how we can develop varieties that might be more regionally adapted. So we obviously have about 15 minutes. So I'd love to open it up to some audience Q&A. Haven't seen anything in the chat, but if anyone has a question and they want to just unmute and just go ahead and start peppering our awesome speakers.
Yeah, I'll start off. Brian Rossnagel from Saskatoon. For Jamie particularly, hey Glenn, how you doing?
Good, mate.
For Jamie, one of the things, just an overall, I make an overall statement first, and that's that I think it's really important for those of us who work in the Montana, southwestern Saskatchewan, southeastern central Alberta, and of things where we do have terminal drought, and sometimes we have what you might call terminal heat, as you say. I think it's really important to remember that in the grain filling process, once the seed starts to form, it's not a zero sum game, fair enough, but it generally, especially with terminal drought, I believe it tends to be a zero sum game. And the first thing that gets laid down in the greatest amount is the protein, and then the starch comes along later. So over my career, I thought about protein first, and then I started to think more about starch deposition and so on as the reverse of what you've been telling us about. So that's just a general comment in terms of working in these kind of environments. And our colleagues in Australia certainly deal with the same sort of thing, just over a different period of time, growing spring crops in the wintertime. But the real question I have for you, Jamie, is do you have any idea about the root growth of these lines?
Yeah,
Is it
so I didn't
better?
have time. I didn't have time to talk about that, but that's a big area of research we're doing. And like I can show you data that shows that there's a correlation in the percent deeper roots with the stay green lines. I think a key thing is with the 6H gene region, which delays senescence, the roots senescence is also being delayed.
Hmm. Hmm.
And so if you can so to put down starch, you have to keep on evapotranspiration. And so you have to keep on pulling water. So you
Right.
need those roots to be active. And so I think that's why the stay green works in our environments is it's extending grain fill um, to allow that starch to be put down. And your comment about the protein first fits really well with that 2H gene region where we can see increased protein. I think we can balance it, but we can see increased protein associated with that earlier heading. I think what we're trying to do, at least somewhat, is shift things, keep the length of grain fill consistent, but shift it a little bit earlier so that we can finish before we have those really high heat events. So that's part of it too. But I don't know, did that answer your question? But we're working hard on this root issue. It's a big focus. And one of the problems we have with that is like we've been trying to do these mini rhizotrons in-field studies.
Yeah.
Those are very difficult to do, and you're getting such a snapshot of the root development that I haven't been satisfied with those results. We have a current project where we're working on that, but we're trying to do some greenhouse studies. But again, in the greenhouse, you're impacting root growth in a different way. So we're working on it. I'm not very satisfied with the techniques we've been using.
Yeah, and it's really, really hard to do that root work in really hard, dry soil,
Yeah,
which is what you're really dealing with.
right.
Um, just one other comment then on that. We've developed these so-called forage varieties here in Saskatoon, things like CDC Cowboy, CDC Maverick, and so on. And I know Aaron's doing some work with some people on that in terms of root development and so on. But one of the things I was quite convinced about when I first found those. They came, interestingly, they all come from 2-row by 6-row crosses, which, as a breeder, you hate to make because it just produces a bunch of crap usually. But those came out and we identified those during the extreme drought of 1988. They were the only things that sort of produced a half-ass yield and looked like normal barley. Most of the rest of the stuff was essentially dead. And I'm quite convinced it's because, for whatever reason, we've latched onto something that has a greater root system and a deeper root system. And I think that's all part and parcel of this stuff. And I know Rich and I have talked about that over years as well with some of the stuff from North Dakota.
Yeah, there's a
Thanks,
couple of
Jamie.
other lines that have been reported to have a bigger root system out of Arizona. Solar is one of the lines. Yeah, so we're working on we're going the same direction there where we really think root system is important. One of the interesting things about that though then is oh, so we have some areas in Montana that have very shallow roots, shallow soils. And so then you're going to lose your effect of the deeper roots if you have those shallow soils. So that's been a little bit of a complication for me as far as, so I have to divide up my environments kind of based on that too, which is more complicating, right?
I might add, I didn't add this in my presentation, but in some of the data that Jamie's got, where you've got those lines that are definitely producing lower protein, and they might be producing lower storage protein as well because they're plumper and you'd assume they've got more starch. Um, there's a set of mutant lines that are missing one or more of the Hordine genes. So these Hordine nulls have been different combinations of the Hordine nulls. And they've been shown through proteomics that where it's missing one or more of the Hordine genes, they're laying down more globulins. So they
Mm,
are still using protein, but not to the same level. And then it's if you've got one mutation, it doesn't seem to have a big impact. But if you're missing the D and a B subunit, then it starts to have a real negative impact on quality overall, particularly extract potential in a fan. Um, so the plant will continue to use nitrogen. It just might allocate the nitrogen into a different protein. So this is something that we haven't really explored yet, and the value of low protein barley and where that protein goes.
yeah, so when I'm making selections, my first selection criteria is kind of for malt is to have a low protein. But then when you start malting it, all low proteins don't perform the same, right?
No.
And so uh and I don't understand that. Like I mean, I don't know why. I just select for the ones that work well. But that's kind of an interesting case. What is different about, so if they're all low protein, what's different about it? And now that we've got this kind of low protein gene fixed, like even having that low protein gene, some of the barleys, they all have the low protein, the delayed senescence, but they all don't have the same level of low protein. Some of them are lower, some of them are higher. And so what's going on there? So there's a lot that we don't really understand. And as a breeder, we just care about performance, right? So we don't, I mean, that's our main goal. And so, but then we have to step back and ask the genetic questions of why. And those are usually slower for us to figure out. It's easier for us to just release something with a performance level than to understand how we got there.
Yeah, and I think that becomes a juxtaposition because you're focusing on yield because we need our farmers to be successful and grow really good, robust varieties. But potentially, then we've got good yielding, low protein varieties. Suddenly, they might not perform as well in the malting or accreditation process
Right,
because we don't understand those nuances around protein quality. It's right protein content, or might be
right.
slightly lower in protein content, but really then the protein composition becomes an factor that might impact performance. And the breeders in the audience have probably had many experiences where they've got really kick-ass high yielding barleys that meet protein quality, but their performance in malting doesn't quite cut it, and they're missing opportunities there. So again, we're probably not diving deep enough into some of those compositional aspects to explain why varieties aren't performing as well. And if we did, then we might be able to design malting accreditation around optimizing malting to see the best in all of those varieties.
Just one thing comes to mind, guys, when you're talking about that. Jamie, I wonder if when you get after you got your low protein lines and you discarded all the high protein ones, and you know they're stay green types, have you looked at the in terms of quality? I know this is very gross and uh, isn't the detail of proteomics and so on, but it's simpler to do. What about evaluating grain hardness or friability on those samples, exclusive of the sort of quotes unquote normal ones, to see if you've got some that have much more easily milled grain, suggesting that they might have more available, the starch might be more easily available. What do you think about that, Glenn?
Brian, yeah, on the merry-go-round again, mate. This has been a conversation we've had for 20 years about understanding the physical texture of the endosperm and how that impacts quality. And I know just seeing some of the names on the participants, I've had conversations with some of these maltsters as well. And yes, they understand that. But if we're taking the big maltsters and it's high throughput screening when they're grading samples and all that sort of stuff, it's probably not totally logistically feasible. But again, we're getting better in the technology we use, and the technologies are better. So again, that's possibly something we could use as a really early screen, because it is both a glucan protein starch combination that impacts texture. So you've got hard grain. It could be one or more of those things that's impacting texture, but that will impact quality.
So what I've been doing early screening is at the early F5 generation, I can actually malt a a small, like a T-ball size, like 5 grams of grain, which I've got at that generation. So for a breeder, one of the issues is having enough grain to do the tests. Friability takes more grain than what I've got at that stage. So I just do that T-ball malting, which is very reproducible. And if I don't, and I don't run all the tests on it, but I run extract and beta glucan for sure. And I get very predictable results out of that, reproducible. So that's my first kind of quality screen at that very early generation before I even have enough grain to do anything else. And that has, so I've got the low protein already. I know that. And then I throw out a whole slew of things that didn't malt well at that early generation. So that's what I'm doing instead of friability. But I think the point I was trying to make is that's kind of easy for me to do that. I can throw those out, but I haven't been taking the time to understand why. Why are those? What's different about those? And if we did that, like if we looked at them more carefully, like maybe it's the protein composition that's different. I have no idea. But I think I'm missing out on some important information there by just kind of throwing all those out.
You can only test so much, Jamie, too. You can't be testing everything for everything.
Well, as a breeder, you just want to get rid of everything that's bad. But to understand for the future, then it's good to try to, yeah, to think about that more. So I'm this conversation, I'm going to think about that. Like, what could I look at to try to, I've got all these populations. Maybe I've got a population that could really help us figure that out.
Maybe we should chat later.
I think
And
so.
you can send me some samples. We put them through some NIRs to get some compositional data.
Well, thanks all. With that, we're right at the top of the hour. So, Brian, thanks for stepping in and leading that kind of ad hoc, interesting discussion to wrap us up. Thanks again for joining us for our third in the Understanding series. Our collaboration with the Brewers Association doesn't have a firm end date, so we're still thinking of ideas and interesting topics we want to discuss with researchers and breeders. And if you have any ideas, feel free to reach out to Wade, Ashley, or me, and we can talk about getting a webinar scheduled. With that, I want to thank Jamie and Glenn again for participating, and everyone else for joining us today.
Thanks, everyone.
Thanks, everybody.